started.
Okay, welcome. The capture will be raised on by before too long. Thanks for coming here. We have almost 90 minutes, I think, for this breakout session, maybe now 80 minutes. So spatial, computational, and algorithm challenge with breakout and Gracewood. I'm Shyam Prabhakar from GIS. And with me is
I'm Grace. I'm also from GIS. Oh, I'm Grace. I'm also from GIS.
And Grace is going to tell you what we're going to do.
Am I?
Session.
Oh, great. Okay, so as part of the spatial initiative here at Human Cell Atlas, there have been a couple of different tracks. And the track that today we are discussing is the computational and algorithms track, specifically the challenges in the computational and algorithm space for building spatial atlases. To start off, just give a brief introduction of what we're doing here. So keeping in mind that the HCA plans to build spatial atlases of the human body over the next 10 years. To implement this, we will probably need to overcome some new computational challenges that we have not, maybe have not even encountered yet. But even without taking that into account, already a lot of the current analytical pipelines face challenges at every stage, from technology-specific pre-processing, so things like cell segmentation, spot calling, and so on and so forth, to quality control, to cross-data and cross-modality integration. So this includes things like integrating with single-cell data, as well as integrating with data sets, spatial data sets produced by other labs or by other technologies, and of course cross-modality integration. As you've heard today, a lot of the spatial technology platforms are not just doing RNA or protein, but both RNA and protein now. There's more and more interest in other spatial modality platforms. And then finally to spatial modeling and analysis, because it's all very nice to collect the data and have these terabytes of zeros and ones sitting on your disk, but we actually want to do something useful with them, and that entails doing some kind of interesting analysis to extract biological insight. But really what the goal of today's session will be is that we want to, as a community, identify the most critical computational and analytical barriers to building spatial atlases, and then to chart a roadmap for tackling them. I think this is meant to be more of a discussion. To do that, what we think that we should do is we want to foster a collaborative community of like-minded researchers who are interested in these problems and who can bring various expertise to the table to tackle these things. So to sort of set the stage, I'm going to start with a couple of questions that we came up with that we want to discuss today. So firstly, what are the most critical computational and analytical bottlenecks that prevent us from building spatial atlases of diverse human tissues in health and disease? So that's a bit of a mouthful, but the idea is that we want to focus on what are the most important challenges so that we can focus our efforts. And also, at the same time, you want to keep in mind the various challenges that come with trying to tackle many different tissues, and also not just in healthy tissues, but also disease tissues. Secondly, we want to talk a little bit about where do current existing methods succeed or fail. So something that comes up a lot is cell segmentation, and how do we rigorously benchmark them? What are the longer term challenges for next generation spatial atlasing? So, beyond the things that we need to solve right now, where do we think the field will go towards, and what are the new challenges we need to address, especially with the rapid technology development? How do we develop community standards for spatial data and metadata formats, quality metrics, and reproducible workflows? So maybe, something that is not as exciting or sexy, but, something that I think is important for people from all different kinds of groups to be able to work together collaboratively. And then finally, how do we build consortia to foster community-driven methods development? Okay, so Shyam, should we just...
A lot of information to digest here. If you could go back to the previous slide, Grace, just quickly. So the key part is this, right? What do we need to build? What do we need computationally to build spatial atlases? That if we want to focus on one thing, because this is actually only a 1-hour breakout session, and some, for everyone to get here, it took some time. We may not get through all of these topics. Let's focus on this. Absolute requirements for building spatial atlases. And why are we focusing on that? As you might have seen today or heard before today, HCA is ramping up its efforts in spatial, and this year we will start some pilot projects for spatial atlasing for a few bio-networks, right? So we want to do it right, and we want to make sure that all of the computational prerequisites are there for starting to build those atlases. And once the data comes in, we have the pipelines to analyze the data. So we took the liberty. So if you can go to the next slide. So we took the liberty of already pre-filling the information, and then presenting it to you for your comments to see if we should remove something, change something, add something. We've charted a 3-phase roadmap, computational roadmap for spatial in the Human Cell Atlas. Phase 1 or step 1 is what we absolutely really need to do early on, rather than after the data are generated, or yeah. So give you a little bit of time to look at this, and then we can dive into it, or maybe we can walk you through. Right. The key thing here is metadata specs, file format specs that are common across various technologies. That's step 1, I think, for pulling in data from various places. And you also need to give the data submitters these file formats and metadata formats. Otherwise, afterwards, if you go running after them to give you more information, it's very difficult. And then another thing, for the panel-based technologies, we have to decide what the panels are for our favorite tissues. So again, that's an upfront thing we need to do even before the data are generated. And then this is just after the data are generated, but still pretty early, right? So the cell segmentation and QC, you really want to have that worked out early on, because everything downstream depends on it. So if you don't even have that sorted out, you cannot move forward. Yeah, so cell segmentation, data QC, and so on. And then integration with single cell data, again, we figured was a high priority early task, because when you generate spatial data, to make sense of it, usually you refer to single cell data. You want to have some simple and some fancy methods for relating it to spatial data to see if you can annotate or all of the cell types that are there in your spatial data, to see if you're finding everything you expect to find, and so on. And then scalability, as you might have seen, a few cm squared of slide area, 500,000, 250,000 to 3 million cells, I think, from one tissue section or one slide. And then you do that across lots and lots of slides, and you've got a huge amount of data. How do we build scalable pipelines for this? And how do we democratize it so that it's not just a tiny number of places in the world that have the resources to analyze these data, right? So this is our phase 1, and maybe we'll stop here and take comments if you have. I know there's a lot more exciting things to be done in spatial omics, data analytics, multimodal spatial integration, and which will all come later, right? But as a step 1 early work that absolutely needs to be done. Any comments on this, thoughts?
On the file format standardization, I think it might be useful to dig a bit more into at which point does it make sense to think about standardization, because perhaps at the raw file, we might want to not ask for standardization, that to make it not just compatible with all the vendors, but also future-proof in case kind of like there might be some requirements, but it might be worth thinking whether there is a common format that everything can be piped through, or whether something like the equivalent of like a spatial data object would be kind of like where we want to standardize. But this distinction, I think, is going to be helpful because if it is before something like spatial data type of object, then designing that common format for the data is going to take some time.
Excellent point. Just as in the case of single cell RNA-seq, right? The different vendors may have their own file formats. So we don't want to interfere with that, but we want something downstream of that machine learning algorithm can analyze in a uniform way. So some kind of adapter between the primary vendors' file formats and to something that all of our data analytics pipelines can read. That's the goal. Does everyone understand what the task is for gene panels?
Shyam, could I ask a
Please.
question? I guess I'm trying to wrap my head around this. For example, the way that we've approached the brain is to say, we have a single cell based reference classification. We'd like to map the distribution of cells across that organ. So now you have a problem. Right? You have a problem. You pick a technology. You're going to map it across that whole organ with limits of that technology. You have a reference to start with. And so your task becomes design a gene-specific panel, scale up your analysis to be able to look across that organ and map it. But that's one version of an atlas. I think what you're asking here is something broader, which is how can we map all technologies using any arbitrary cutting through, say, classify what we're talking about. Atlas. Or are we talking about a set of methods which will be generalizable for spatial technology, anyone in the world?
I think we envision multiple technologies for one atlas.
One atlas built of what? Democratized data from whoever? What does it mean?
Jasmine, you have a comment?
Right? And don't have a FASTQ file, right? So the output of that, right, just based on imaging 1,000 genes, that amount of data gets compressed and we don't actually have the pixel level information. That's really what you need. You need the pixel level information for every prior to decoding of the genes. And how does one envision that when, VisGen doesn't come off with like, you get a QP-TIFF from Codex, you get, right? So we have to think even prior, even just saying generalistically that there's this midstream way prior to spatial data just doesn't yet exist. And there doesn't seem to be yet a path, which is why we're in the room, but just putting them as bullet points is not really achievable, right? So what Ed's asking is very clear. Do we take the atlases that we're making and try and standardize the acquisition based on the technologies we have? That's likely more achievable, right? Because, as to the first question and the second question, they come along the same lines, is that even getting to spatial data, if we haven't yet first set up the standardization of if you run a VisGen experiment or a Xenium experiment or et cetera, those outputs are never going to be, they're different levels we're not capturing pixel. And no, there's nobody really high leadership from companies here to hear that, right? Which are—
There is some leadership.
Oh, okay, here. If we can have there's Serge, I see Serge. We need I come from genetics. We used to keep BCL2 files, right? Prior to them being FASTQ files. And that's the place we're in right now for spatial omics, I think. So there has to be, to Ed's point, it's a really important point. Right now, I think we can go from an atlasing perspective and kind of utilize across the technology something that becomes standardized into a spatial data. But I think it would be a dream if we had a FastQ, but we're not there. And I don't see, based on the level by which we capture these off the machines, it's not achievable.
Maybe Grace, you want to comment before I come in?
I'm trying to maybe if I write that down correctly, you're saying that the raw data right now is too variable and too raw basically across the technologies. Mm-hmm.
Yeah. So coming back to that analogy, we don't keep BCL2 files now, right? So what do we want to keep, and what is the file format for that for spatial is the question. So that's what we try to capture here. We don't want a shared file format for the raw data. Does that make sense, Sun? Yeah, it's not raw. By the time you're not, some vendors, they won't give you the raw data of the machine. You get the process data, right? Which is okay, but that's all you want.
Because if you get, so for example, you have cell segmentation changing, like, I mean, it's a never ending wheel, but unless you contain some version upstream of that, you're beholden to, because it's an image now, right? It's not that you put it in a droplet and got off a sequencer what was in the droplet, right? So that doesn't come off. I have most of the machines, just by the way. So I know the frustration of all the different formats. And as to Ed's point, we want to utilize Alan Brain's VisGen. And he's saying this specifically because that file format is they keep a little bit more upstream, is what I'm saying, than maybe another format, right? And so how do you standardize that in an imaging-based thing where we're not keeping truly the pictures, right? Because that data, no
We're not
offense,
keeping the pictures. Yeah.
right?
So one way you could do it is, like you said, cell segmentation keeps changing, right? We don't want to get too much into implementation details, but I guess you're raising an existential question, like, is it even possible to have some shared file formats downstream of what people provide? One strategy is for these imaging-based technologies, you keep the spot locations. So this RNA, this XY coordinate, this XYZ coordinate, and then you have some representation of the cell segmentation masks, which can change. And then you're covered, right? So I think this file schema, or whatever you call it, covers a large number of technologies. And then you can have another one for these grid-based or probe-based, sequencing-based spatial technologies. So we can have two sets of file formats, for example, right? One for imaging-based, one for sequencing-based. But we do need to have some kind of standardization so that we don't build pipelines for every single kind of technology separately. Does that address your question, Ed? And I think there was another question also in there, or was that? Oh yeah, a mapping from single cell references. Yeah, so I think we want to enable both directions. We want to say we have spatial data, we have a single cell reference, we want to do label transfer onto us, take our single cell labels, transfer them to spatial. But we also want to de novo discover labels from spatial and then identify the best corresponding single cell label from that. So we want to go in both directions. That's in our phase 2 or phase 3. Thank you. Yeah. I think that's very, very important. Yeah. So that's part of this. This is a very broad umbrella, integration with single cell data. So it includes what you suggested. And Chloe and Gary talked about CAP, the cell annotation platform, today. So that will hopefully be our reference for each of the bionetwork atlases. And so the CAP annotations will hopefully be integratable with spatial data and that'll be one of our tasks in the computational track.
Maybe I can also add that on a niche level, there would be a need for having the human cell atlas be a reference, not just on the kind of.
Absolutely. We'll come to that. So niche determination or niche discovery or whatever is a major task. Perhaps not phase one,
Yeah.
so more in phase two. So are we done with phase one? Any other comments on phase one? Anything to add here?
I guess the idea here is to define the things we need to define so that the people can start generating their data.
And QCing them.
And QC them. And then so far, everything else is on the next stage. But of course, I think open to discussion on what people think are the
Is there some
priorities.
font issues? Some of the last row is
Yeah.
disappearing.
Well, I'm trying to take notes, but badly.
Oh, okay. There we go.
Yeah.
Yeah, so integrating single cell data is certainly in the P1 priority one area. Oh, we do also want standards for what is a good spatial data set. And that's going to be challenging also for these many different technologies. But you might have seen everyone who presented today, they presented some kind of QC benchmarks. And those are the obvious ones we want to put in. We want to have benchmarks not just for sensitivity and specificity in calling RNA molecules, say, but also for cell segmentation, which is less straightforward, but a big issue for spatial omics data. And then if you have a bad field of view or something, so you want at every level, right? You want QC metrics for spots or probe sets. You want QC metrics for cells. You want QC metrics for fields of view and QC metrics, of course, for the entire data set or entire tissue section. Those are fairly standard or well established for single cell data, not so well established for spatial data. Okay.
I think there's a few comments in the audience.
Comments? Yeah.
I wonder if it would be possible to recommend to everyone that is generating data to acquire paired H&E to the best of their ability, even if it is on a consecutive slide. It will definitely help with the integration down the line, because I think after all the data sets have been collected in individual manners, integrating across groups, and then kind of like having the conversation of what is the integrated spatial reference of the human cell atlas, because of some of the challenges that were already discussed, perhaps H&E is a modality that everyone can standardize around.
Thanks for that. So there's many ways we're addressing H&E. So there are actually 4 tracks of this spatial task force. We are only track 2. Track 1 is the technology track. So Holger's leading that, Holger and Jay. With track 2, computational. Track 3 is the portals, I think. Yeah, portal development. And track 4 is atlasing. So Chloe is leading that. So what you suggested is really quite relevant for track 1, which is, H&E is one of the technologies they should be adopting. And also relevant for track 4 that, you want to apply this in all your atlases. And then for us in track 2, the relevance of H&E is more like, okay, what are the computational tools? So we're not going to make recommendations on what data they should generate. But what we will assume that they will generate H&E data, paired H&E data, and say, okay, so what are the tools we need for that? And we'll come to that in one of the succeeding slides. Anyone else on this, Adai?
Sorry, I had a follow-up comment to that, but it was along the same logic as the multimodality, right? So kind of image registration, right? Is it landmark based? Is it those kind of things? Because that ties very streamlined in from track 1, right? So we have to be thinking about protein H&E and RNA, if that's the case, then, I mean, the easiest one right now is landmark registration, but kind of first prioritizing that in the beginning, because that is going to be agnostic to cell segmentation and those kind of things. And if we get that right in the beginning, we can fix cell segmentation. And it's highly applicable to all the AI tools downstream.
Great suggestion. I see Grace adding image registration, I
Yeah.
think, somewhere.
I think
Yeah.
that's a great idea.
Yeah, that we need upstream in step one itself. Yeah.
So I guess you've been talking about Atlas as in the data that we're generating in-house or from public domain. Because one of the issues we have is when we look at public domain, it's usually missing for a lot of metadata. Even a notation of cell types is missing. I'm just wondering whether it's possible. Some kind of checklist that people can go through and say, yeah, I provided this. And where it's provided at, its geo website supplement. I think the problem will become bigger for.
Yeah, thanks for that key issue. In the single cell atlases and the human cell atlas, some of them were developed by the HCA bionetworks, some by the wider community and just brought in and integrated after publication by the bionetworks. And then exactly the problem that you mentioned came up, which is you're reading the supplementary material of that paper, you have very little fuzzy knowledge of the metadata. But then usually what the bionetworks do is they chase down the authors and keep emailing them and get the information. But for people submitting directly to the HCA data portal, we now have a metadata spec. We have a tier 1 and a tiered metadata spec of open metadata, access control metadata that's recommended by the HCA. And the data wranglers request that. So when either data was submitted, we submitted the metadata in the HCA requested format. And so that's this part. So we do want metadata specs for spatial data right at the start. I think that's a great suggestion. Once we have a metadata spec ratified here, we can recommend it in the checklist for journal editors. Any other comments on phase 1 back there? Yeah, there's a microphone.
Could you please turn the font size bigger because we couldn't see from the back?
Is that better? Sorry. How do I do that? I just break it down to 2 slides.
How's that? Is that visible now? Okay, great. Okay, Bridget, where? Oh, did we lose Bridget? Yes, we lost her. Okay, let's move to phase 2.
Sorry, let me change the font size.
So step 1 in phase 2 is democratizing access. There's something that we don't always think about, but we're focusing on it increasingly. And what that means is that a large number of members of the HCA community should be able to access the data and analyze the spatial data, because spatial data are huge and computationally hungry and memory and storage hungry. So yeah, how do we make sure that we lighten that load, right? We make our methods less resource intensive, or we provide servers where people can upload their data and get some basic analysis results. Either way, there are many ways we can address that issue, but this is really one of our major tasks in the HCA. And then democratizing all of this, and then this is the stuff we want to democratize. This is all the fun stuff, all the fun algorithms that we like to develop for spatial. someone mentioned niches, so that's in here. We talked about multimodal, that's in here too. And integration with H&E, that's there too. This is predicting spatial omics from H&E, but we can, and this is integrating with H&E, so it's all covered there. Yeah, this is a more disease-focused angle. For single cell biomarkers, it's mainly, okay, what are your pathway-associated genes? What are your differentially expressed genes, cell type specific genes, and things like that. Various kinds of regression analysis, you can say. For spatial, you want to find things, you could just use single cell algorithms for spatial biomarker discovery also, but you want to use the XY coordinates as well, right? So it should not be so trivial. There should be new algorithms there for spatial biomarker discovery. Okay, I'll let you take a look at this, and perhaps people can comment. Is there something that is a solved problem, so it really should not be in this list? Because this is just about things that we need new algorithm development for, new pipeline development for, right? Do we need to delete something here, or something missing that we need to add in phase 2 to complete our 2D initial spatial atlases?
It's a question like, do you see this as sequential step between phase 1 and phase 2, or you see them as like the in order priorities? They may be happen in parallel, but some analysis methods may happen first because they are essential, and some may be happen in parallel because they are interest to certain groups.
So the way we envisioned it was sequential, meaning temporal ordering rather than priority ordering, because these are also extremely important, but they can happen a bit later. Whereas the stuff on the previous slide has to happen right at the beginning.
So like, do you have expected outputs from the phase 1, and then do we need that outputs in order to input to the phase 2?
So there's no dependencies like that, at least that I can see. Or to put it differently, phase 2 can start at the same time as phase 1, but phase 1 should not start after phase 2, right? So phase 1 has to start in the beginning, and obviously different teams will tackle different challenges. So while you are tackling phase 1, someone like Jasmine can tackle phase 2 or something like that, right? That's fine, but someone has to tackle phase 1 early on.
Yeah, so I can maybe add on the defining niches compartment that it would be great to have kind of a query component to that, where there are some open questions about how to compare niches, say lymphoid aggregate structures from multiple types of tissues, I think, and that could be one component where the human cell atlas could be a reference. So sort of being a reference of niches, so moving a little bit away from just cells, but also having this kind of concept of a niche reference.
That's a very nice suggestion. Yeah, thank you for that. I think Grace is writing it down. So niches or spatial structures, for example, how are you define it? Some kind of spatial objects or spatial patterns, you can say. And niche is an example of a spatial pattern. We should make that queryable because they could be shared across different data sets for the same organ, or even shared across organs. Like you're talking about lymphoid aggregates, for example.
I mean, it's not very priority, but so from my perspective, I do the data, like the biologists send me the data, I do the data analysis, I can create the UMAP, give them all the differential expression, but like at the end, they need to be looking at it and then telling me, yes, this is this cell type. But what we are facing right now is if I have multiple slides, how am I going to give them in an interface that they can see all slides at the same time? And then, like, for example, when they write a gene, they can see it all together. Like, we go through different, like, kind of vendors, ask them. Nobody has a solution about that. So if I don't know, anybody knows, I will be very welcome. But if the Human Cell Atlas can provide something like that, which today we see the cell type, that's very great. But like, if I can have my multiple slides in one go and then show them, and then we can see where it's located in the tissue, disease control. So it will be very perfect. Yeah.
That's an excellent point. The single cell equivalent of that is a lot easier. You just have a heat map where each column is a data set or each row is a data set or something like that. In this case, you can put each slide as one rectangle on your page, but yes, yeah. So I think visualization becomes a more interesting question. So perhaps Grace, we could add that. Yeah, okay, you've added that already.
Yeah, so I'll just comment that this has also come up in the data portal track, how to visualize things and how to query things. And I think they probably need to work closely together to try to figure that part out. Because I think in general it's quite poorly defined right now. But yeah, super helpful feedback.
Yes, there's some tasks shared across tracks. The metadata format is also shared across the tracks. I think we can go beyond 6:10 p.m. We were supposed to stop at 6:10 p.m., but everything was running late, except we have lost Bridget, so I don't know how long we can run. Dane, would you be able to track down Esther or Bridget and ask them when we need to end this session? Thank you. They'll be at the registration desk. Any other? Oh, this looks like a very uncontroversial, like everyone is agreeing mostly. Any concerns? Maybe then we can, okay, let's talk about if no more questions about phase 2. We can talk about longer term objectives, which is spatiotemporal modeling, not just static snapshots. Spatial trajectories, just like we have pseudotemporal trajectories right now. And then additional modalities we're not talking about. Right now we're talking about H&E, protein, RNA, but we will get to spatial genetics at some point. And then spatial chromatin profiling is already there, but not so well established in terms of commercial vendors providing solutions. And then, of course, we heard about Stellaromics 3D spatial atlasing today. And I think Visgen also talked about, since they have clearing, you can just take more Z-planes, and that's some amount of 3D. So yeah, we do want to move to fairly quickly to 3D atlasing as well, rather than registering 2D images together. Any comments on long-term objectives beyond the first pass V1 spatial atlas?
Question on the spatial temporal modeling, because most of the technologies that we have discussed so far are invasive. And so if we are talking about collecting samples from human patients, what are we thinking in terms of being able to have different time points? Are we going to use different modalities, or are we going to use different species to try to fill the gaps?
I guess how do we get temporal information from humans? Yeah, it's a good question. I don't know the answer. But I guess, I mean, you do have pre-post, but that's not really temporal. It's just 2 snapshots.
I mean,
it's not dynamic. Yeah.
Exactly. You can get it with different modalities that, I don't know, like people can do MRIs. Is it something that we want to, like we talked about standardizing around H&E, which is probably the most commercial, the most viable solution, but would we also want to think about when we go multimodal, one of these modalities that is non-invasive, and then having kind of like pair data when possible to be generated to then us on the computational side, then we could build imputation methods.
That's a very cool idea. I definitely well deserved in the longer term objectives. To think about modalities that are not necessarily molecular or traditional pathology type imaging, which can give us dynamics. I don't know, anyone else have any ideas about what kind of spatial data will allow us to infer temporal trajectories?
It's for medical imaging data, for example, CT, but the problem is that the resolution is relatively low.
Yes, yes.
You perhaps have to have some training data with paired pathology image to link with the spatial genes.
Yeah, so I think you're both saying roughly the same thing, that you have some less informative data type which you can take in real time. I'm not an expert on this, but I imagine fMRI can give you some amount of information as a person is behaving. But yeah, and then some training data perhaps to fit that to the spatial data.
Yeah, I think along the points of spatial temporal modeling, we could also consider growing organoids from the same patients and then give the same stimulus, but then you would sample it from different time points. And you sort of at least have an anchor that it comes from the same donor. And you could do some modeling if you find like the same niches and see how they would evolve over time.
Thank you. Yeah. So essentially creating multiple instances of a system and sampling them each instance at a different time point. Yeah, that's the standard time course strategy, which you can do for model systems. So a model system being either your or 3D organoid or mouse, something like that.
I'm wondering, is it possible to build a universal coordination system so that when we collect some slides and we know this cell is located at a specific location so that we can compare across different technologies and data sets?
You're referring to like a common coordinate framework, right?
Sorry?
Like a common coordinate framework, is that correct?
Just like a map.
Right.
Yeah.
Okay.
Thanks for that. I think HubMap calls that a common coordinate framework, if I remember correctly. And they did do a fair bit of work to develop that. And you want to have the sampling to be able to justify that framework, meaning you want to be able to sample all. So Ed mentioned it too. Ed Lane mentioned it in his talk. So I think they used for brain, they did use a common coordinate framework to map all their pieces of tissue into a whole 3D landmark system. And we do hope to leverage the HubMap common coordinate framework over here.
So this also came up during the data portal, and maybe for discussion, was the idea that pathologists and doctors have in general already defined a common coordinate framework. We just don't use it right now, and maybe we should think about that.
Are you talking about those plastic models I see in the doctor's office?
Exactly. Was it? That's all. Sorry, I think that was a comment from, yeah.
My question is not about phase 3 or phase 2, or much more, probably it's phase 0. Does this community or the group have like a common list of benchmark tools that the rest of us could use for doing most of the spatial analysis, right from data QC to the most advanced analysis? Is there a recommendation list from this community?
Good question, but I see people looking at their watches. Just to be clear, we can go till 6:30 because we're running late. Is that correct, Dane?
Yes, but there's a reminder that there's a closing talk at 6:30 that you're encouraged to join.
Okay, so we need to get to the main auditorium by 6:30. Is that right?
Yes. Yeah.
So then we'll close here at like 6:25 or something like that. Or let's say 6:20 even. So yeah, we try to. I think long ago in the Human Cell Atlas, there was an attempt to provide some reference algorithms and data analytics pipelines, but no one could agree on what those would be. And anyway, it's hard to get data analysts to all use the same pipeline. So maybe it's better to just not have a preferred list. Because that gets controversial and there's no real good way to say this. There's too many conflicts of interest also there, to be honest, when we try to recommend that.
Maybe not recommending tools, but recommending tasks. Like which tasks would we want to see the results of the benchmark so that when people are developing new algorithms, they know these are the tasks that would need to see results on for it to be considered to be used for the next generation.
As in a set of benchmarks for quantifying performance of the algorithm?
It's kind of like, do we want to see things like recovery of cell types, and we would quantify that against the reference atlas of the human cell atlas, or would we want to see? I don't know. I would have to kind of like think about exactly what the tasks would be, but then we would not be prescribing any of the how to do it. Just this is what we want to see how you do. Yeah.
That's excellent. So that's part of our phase 1 actually, which is benchmarks or spatial. Okay, I guess in phase 1 we talked about benchmarks or QC metrics for data. Somewhere in there, Grace, there is benchmarking also, right? Somewhere in these slides.
Yeah, I think we said the word benchmarks, but we didn't put it down as a task. But
Okay,
maybe
great.
so establish benchmark data sets and tasks that people can use for algorithm development.
The reproducibility of the algorithms can actually be measured.
Got it. So how do you benchmark the key algorithms for the various stages of the pipeline? Yeah. Even that gets tricky. So at least in our experience, what we found for single cell RNA-seq, there are some published benchmarking strategies. But the problem in single cell is once you generate the RNA-seq data, you've destroyed the cell. So you don't know the ground truth of that cell. And no amount of cleverness in algorithms can get around the fact that you don't know the ground truth. So how do you benchmark an algorithm that tells you what that cell type is, right? When nobody knows. Independent modalities help. So that's one way of benchmarking. So your single cell attack-seq and single cell RNA-seq clusterings, agree with each other when you have multimodal data, things like that. So yeah, I think it is valuable to define some benchmarking strategies for the algorithms.
Would it be useful to have a kind of a challenge, like a dream challenge with these tasks, just to direct
yes. Thank you.
biology community?
That comes to our implementation strategy. We do want to organize hackathons for our key challenges.
Just add on about the multimodal data. So because the samples, most of these samples, I believe, will be from clinical or from hospitals. So would it be, I think it would be useful to have like some of the medical history, medications that is that the subject is taking, some of these medical history.
See, so clinical metadata, like medication history, diagnosis, drug response, things like that. Yeah. I think it's a good suggestion. We can put it under metadata specs.
Just if you are thinking in long run, in future, if you are to use this as a control or some of the reference data to compare to the disease one, and then this clinical scoring will be very, very crucial for comparison.
Absolutely. When you want to do biomarker discovery, you want to have as much clinical data as possible. So maybe some basic essential clinical data we can have specs for in our metadata spec. And so we kind of reached the end of this time slot. I hope to make all of this a reality. I'm really hoping to draw on all of your expertise and willingness to start creating algorithms and pipelines to address all of these challenges. So please reach out to Ollie Stegler and myself and Grace and other people on the computational task force. We are going to, amongst ourselves, start forming teams to address some of these challenges. And I think they're very important and exciting problems to tackle. So hope to have all of you with us on that journey. All right. Thank you so much, everyone. And back to the main auditorium.
No problem. The computer hates me, but hopefully they don't get distracted by the donut wall. There's a donut.
There's 2 donuts.
Hi.
Benchmark. Can you add to consider a generation of a synthetic data
Generate.
set?
Okay.
Because I fully agree with Shyam's point. We have no ground truth in single cell, but a synthetic data set. If the community could agree on a synthetic data
Is
set,
that-
that could be.
All right. Thank you.
One of the technologies